So I'm going to go ahead and start that recording now. Again, this is Jeff Wuchich, co-founder and president of CureHC, and with me is Simon Frost, who is a fellow board member of CureHC and is the mastermind, one of the masterminds behind this AAV-mediated gene therapy for AHC. Welcome, everyone, and I will turn it over to Simon now.
Great, thanks so much, Jeff. So I think I'll probably start just by saying that this has been a massive collaborative effort, not only by the board members of CureHC, but by various foundations across the world, really. And this has been a parent-led initiative, so huge collaboration across parents, but also across scientists. And I'll get to all of that later on, but we have some of the best scientists in the world in this field that are behind this and are helping advise through this process. And we have built what I think the general team believes is the best approach to a targeted therapy for AHC that we might expect for the next 5 to 10 years. So that, hopefully I'm not overstating things, but I'll get through some of those thoughts in these slides. I'll start with the disclaimer. So I'm not a doctor, and this is my very bad attempt at a joke, but my disclaimer is that I am a parent, and I care very much about this disease like all of you do. So the first slide talks about why AAV-mediated gene therapy. And the major reason we wanted to do this is because after very much research, we ultimately settled on this being the best approach for our children, and especially for children that are living with this disease now. There are a lot of ways that we could go to find, for instance, a CRISPR-Cas9 approach to therapy, which is genetic modification therapy, gene editing. But that right now is very risky and probably won't be able to be delivered anytime soon to our children without high levels of risk. What we did as a team, and one of the very first things I did when my daughter was diagnosed, which was October last year, was to hire a scientist who worked at the NIH. Her name's Natalia Morsch, and she is on the medical advisory board of CureHC, and she and I have worked very, very closely for a year now. She's pretty much worked full-time on what you see in front of you, which is understanding the structure and the function of the protein associated with this disease, looking at the pathogenesis of the disease, how the symptoms come to effect in the disease, how they show themselves. The symptoms versus the mutations and how those are correlated, and then looking at on-target therapies. And by on-target therapies, I mean addressing the problem, not trying to mask the symptoms. So those are the things we looked at, and we looked at them for, it's been more than a year now. It's been a year and 2 months to try to analyze the best approach. And we really thought there were only 3.
Simon, I'm sorry to interrupt for just a second. You seem to be breaking up just a little bit as we've gotten going here. So I don't know if you're on a headset or a mic. You might want to do a quick mic check.
Yeah, let me dial in. Maybe that would be better quality sound.
Okay.
Sorry, Jeff, do you have the number for the dial-in?
I do. Stand by. The dial-in for the number is 872-240-3311. And then the access code is 335 970-469.
Sorry, Jeff, could you give me that access code one more time?
Sure, 335-970-469.
Jeff, I think I'm muted.
I think you'll have to mute your computer. There we go. I think, though, if you used your particular attendee code, that might link you to this and to the computer, in which case you might want to just dial back in without that attendee code, and then we should be good to go. I'm sorry, everyone, for the slight momentary technical difficulty, but we do want to make sure we get this as clear to everyone as possible.
Hi, Jeff.
Yes. We still have.
To how to mute one thing and not the other.
Okay, well then I guess we'll just hang up and have to make the best of it, and just keep going on the computer part.
Okay. So what we're looking at here is the slide showing the protein associated with the disease. And this is what we're trying to target. So this is the sodium-potassium pump, and you can see it's a very complicated set of biological machinery. On the right there, you can see it at the molecular level, and on the left, you can see it at the structural protein level. And this protein changes shape, and it changes shape very often. The idea here is that it's very hard to treat a protein like this with drugs or small molecules because it is changing so rapidly and has many different conformations. And drugs is one of the three ways that we looked at potentially treating this disease. We know that drugs like flunarizine have off-target mechanisms for helping with the disease, but not on-target mechanisms like making the protein work better. So drugs was one option. AAV-mediated gene therapy was another option. And then gene editing like CRISPR-Cas9 was the third option. CRISPR-Cas9, like I said, seems like it's a little bit risky right now. And AAV-mediated gene therapy seems like it has been very successful in other diseases. So that's the approach that we followed. So AAV stands for adeno-associated virus. And this slide says, what is AAV-mediated gene therapy? So the idea here is that we take this virus, and you can see what the virus looks like, and it actually looks like what we have depicted there on the left. It encapsulates DNA, and it encapsulates our gene, which is ATP1A3. And we deliver that gene inside the virus to neurons that are affected by the disease, so that we can add functional copies of that gene, which will form those proteins that I showed you on the last slide, and mitigate the effect of the proteins that have been badly formed by these gene mutations. On the right-hand side of the screen, you can see a big blue arrow, and that is showing the adeno-associated virus. and it's showing the adeno-associated virus next to an adenovirus, which is a much bigger virus, which is more dangerous and carries more DNA. So we've got this tiny little virus that is carrying our payload, and our payload is our therapy, which is a functional copy of our gene. Here's another depiction of it in a more graphical form. You can see we've got this virus, which is that helix-looking shape, and inside of it is a strand of DNA. That's kind of how I think about it, the virus encapsulating the gene, which would then deposit its payload into those neurons, as I said. So packaging DNA in a virus. There are various different forms of AAV. All of these have been generated to go to specific parts of the body and express well in those parts of the body. You can see the one that we've chosen for our therapy is AAV9. And AAV9 goes to all the parts of the brain that we want it to go to without transfecting other, the heart, the lungs, the kidneys, and other organs of the body that we don't want it to go to. So
yeah, in other words, it's targeted just to be at the brain and nowhere else.
That's right, at the brain and at specific parts of the brain that we really want it to be in. So inside, we have the right virus to target the right cells. And this shows you where we're going to be injecting. So, and it shows some of the issues that we're facing with AHC. So AHC heavily involves the motor loop, and you can see the motor loop shown there. A lot of problems seem to originate in the cerebellum. There are a lot of cerebellar symptoms to AHC, like ataxia and dystonia. And those, we're injecting in what you can see there called the cisterna magna. And we're injecting in mice starting on November 26th into their cisterna magna to see whether or not we can get expression levels across the brain. But the idea is that symptomatic episodic events that are caused by AHC, we want to prevent, and we want to inject as close to the cause of those episodes as we can. And in this case, of course, a group of scientists that decided that injecting into the cisterna magna, which is into the cerebrospinal fluid, very similar to a lumbar injection, is our best approach to this therapy. So why is it a good option for AHC? It's targeted. We can inject in the right places. It's functional. It's providing a functional copy of a dysfunctional gene. It's proven. This has been done before using a different method that we can't use in humans, but using a method that Dr. Steve Clapcoat of Leeds University used in mice to rescue the symptoms of AHC. And it's also been proven across multiple other diseases. And it has a low overexpression risk, something that all of the community worries about, is toxicity associated with delivering too many genes or too many proteins to specific cells. So I'll stop there. There's a lot that I just delivered. Any thoughts or questions?
I know, Simon, you mentioned that it was similar to lumbar puncture. Maybe you can also talk about how it's a little bit different.
Sure. So lumbar puncture is delivered into the base of the spinal cord. This is delivered also into the spinal cord, but much higher up, right at the top of the neck. And it's into a cavity that is quite a large cavity called the cisterna magna. But that cavity is right next to the cerebellum in the midbrain. And the idea here is that if we inject large quantities of virus carrying these strings of DNA, the first place they're going to go is to those two regions. And if we were to inject into the spinal cord lower down, we'd probably get quite good levels of expression in the spinal cord itself, but lower levels of expression in the cerebellum and the basal ganglia and the midbrain, which is really very important areas to transfect.
Right. And we do have a question here that I think, all right, we have a couple different questions here. Does AAV therapy impact only neuron cells or all brain cells?
Yeah, that's a great question. So you can target both, and you do that with different promoters. So in our case, we only want to target neurons. We don't want to transfect glia. We have the ability to transfect glia using a different AAV capsid and a different promoter, but we've chosen not to because ATP183 doesn't express in other cells, it only expresses in neurons. We don't want to give ourselves more risk by transfecting cells that don't normally express this gene. But for instance, maybe this question is related to ATP182. For ATP182, we can target the glia and express ATP182 in those glia so that we can potentially correct issues with ATP182 mutation.
Right. So I think our idea would be that if this is successful as is, we would design a separate study to address those potentially. I think is the point to drive home on that. We also have another question which may dovetail right into your next section here, Simon. How will we know if it's effective? What sort of markers are you going to be looking for?
Absolutely. So there are various levels of markers that we're going to be looking at. The first is that we are, right now, the first test we're doing, which is November 26 injections. We're injecting mice that don't have the disease with 4 different types of vectors. The only real difference in those is what the promoter looks like. But we are analyzing where in the brain those viral vectors are going, how well they are transfecting cells, how many neurons they are delivering their payload to, and where those are in the brain, and how many proteins they're producing, because we want lots of proteins to be produced that are new functional proteins. We're testing where and how many of those there are across multiple regions of the brain. So the area of the brain that coordinates motor function, the area of the brain that coordinates memory, and then the various other parts of the brain that help with those functions across the whole motor loop. So we'll be looking at cells, especially high frequency cells, to see how well they're transfected, and then we'll be picking an AAV serotype. We'll be picking the one that works best to deliver it into a disease model, into a mouse colony that has the disease, and then doing a battery of tests on those mice to see whether or not their cognitive function has been restored, their ataxia has been abated, their dystonia has changed to better levels, and then their memory has improved. So those 4 things are things we're going to be testing heavily in diseased mice through behavioral studies, and that should kick off in April.
All right, and the only other question we had that we haven't quite addressed yet are what are some of the risks to this type of injection into the cisterna magna?
Great question, and I'll get more to it in further slides. But anytime you're injecting into the cerebrospinal fluid, you have risks. So you have risks of infection. You also have high risks, which are mitigated early. but risks that you're going to get an immune response. So a lot of kids have a natural immunity to these types of viruses. We'll be checking that in their blood before they're ever injected, but immune responses are not good in this case. We don't want the body killing the disease, but we don't want killing the virus. And we also don't want the virus to cause sickness. So that's another big risk. But I've got a whole list of risks at the end of the set of slides that I'll go through.
Okay.
It's not exhaustive.
Right. With that in mind, we might want to go ahead and move on. But these have been excellent questions, and please continue to use that chat box or the other means that we talked about to get those in. Thank you, Simon, and please.
Yep. The next thing is, I asked the question, has this been done before? And yes, it's been done very successfully across multiple diseases. It's also been done in certain diseases that it has not been successful in. But for the most part, it has not been a high-risk treatment. There's no known effect of delivering AAV9. It's not inherently harmful, but it's not always helpful. There are some very good cases where it is helpful, and I think that our disease sets up well for being a success, but we still need to do the tests. This is a therapy that's been around for a long, long time, and for various reasons it has not become mainstream. But the most successful examples of AAV-mediated therapy come from patient-led initiatives where parents are driving the research. So I think some good examples are Batten disease, spinal muscular atrophy, Duchenne. These are very good examples of patient and parent-led initiatives that have been very successful in their clinical trials. I have a slide here talking about the success, and one big success recently is inherited retinal disease. The drug, which is Luxturna, is almost identical to what we're doing. So it's taking AAV2, it's packaging a functional copy of the gene into AAV2, and then it's directing it, it's injecting it directly into the back of the eyeball. And you can see a graphical depiction there of what is done in the therapy. And I have a video here showing exactly how it worked. It doesn't seem that the video is working very well, but we can share that with everybody after this call, so that you can see in this video how folks have gone from being relatively blind to being able to see and walk through mazes without any assistance. Another very good example is spinal muscular atrophy. In spinal muscular atrophy, typically kids don't live, especially with SMA type 1, they don't live much longer than 10 months. In their trial, which involved almost identical to what we were talking about, AAV9 capsid with an enhancer and a promoter and a gene included in that capsid, which is very similar to what we're doing. It took the survival rate to a very high level. So there were a certain number of patients that were injected, 24 or so, and those patients, 15 patients, and all of those patients survived significantly longer than the average. And a year after the trial, every one of those patients had not passed away. So a highly successful trial, and you can see here all 12 infants were still surviving. 6 out of 7 required no feeding support. 8 out of 12 were able to speak and sit unassisted, and 2 were walking. Unprecedented results for that disease. Another good example is gangliosidosis, complete correction of enzymatic deficiency in that disease back in 2007. I have another video here which you'll be able to see when we share these slides, but it's a video of cats that are untreated, that are unable to walk, and then the treated cats are running around chasing after laser pointers. So it's a huge difference between untreated and treated cases. So highly successful in that disease. Wanted to pause for some questions.
We don't have any other questions right now, Simon, so we can, I think, press on, and certainly we'll have other opportunities, but keep those questions coming if you have any.
So are there risks? And I identified the ones that keep me up at night. I didn't identify all the risks here, but I think that maybe some of these are a few that you've been thinking about too. I worry a lot that it won't work. We may not get adequate expression and biodistribution. It might not get across the whole brain. It might not cause sufficient proteins to make a difference. That's one of our risks. There's a risk that AAV causes that immune response that I talked about before. I've put there are no known health hazards associated with AAV. It's, as viruses go, it's about as safe as it gets. It's not known to cause direct diseases in humans, but there have been certain cases where there has been that genetic editing effect of CRISPR-Cas9, where a gene inserts itself into the genome. But that has been sparse enough and has been at very, very, very low levels where it should not cause major harmful effects. And then there have been possible connections to cancer, but mostly that's been refuted. The risk of intracisternal injection. So like I said, any injection into the CSF is risky, especially when you're injecting so close to the brain. And that was one of the differences between the intrathecal injection and the intracisternal injection, that we are going so close to the brain. The immune response is a risk that I think we can mostly mitigate prior to injecting into humans. And we can screen by child. So if a child has an immune response, we'll know that before injecting them with the virus. Toxicity, so at very high doses, especially if we're injecting intravenously, which is not the route of administration here, we're injecting directly into the CSF here. But if there have been cases of severe toxicity at very high doses of virus in intravenous delivery. But with the CSF delivery, we can get a more precise dosage. And those dosages are well established across multiple diseases, and we'd be following paths that have been proven to be safe. Again, though, there is a risk. And then anytime you're doing something for the first time in humans, there's a risk. There is not always a good correlation between what happens in mice and monkeys and what happens in humans. So chances of success, I've put there, we don't know our chances of success until we try it in mice. At this point, it's a very subjective guessing game as to how well we're going to be able to treat this disease using this approach. But the best we can come up with objectively is to say that we think that this is a very good approach using various hypotheses on performance of the viral vectors, the likelihood of our gene to express, and the results if it does. So we think that it has a reasonable chance of success, but putting numbers on that is impossible to do right now, and nobody will do it until we got through the preclinical research. There are 3 phases here: preclinical research, clinical testing, and then the rollout of the drug, and the drug being the virus encapsulating the gene. Right now, we're almost through 1.5 stages here. So we're through the lab research for the we're through a lot of the lab research, and we're starting to inject animal models of the disease. And we've designed our experiments in animal models, in mouse models of the disease. So now it's really just about the execution of the preclinical research. We are working with Dr. Steve Gray, who is an expert in AAV-mediated therapy, and I'll talk a bit more about him in 5 minutes. And we're working with the Jackson Laboratories on animal testing, who are one of the biggest mouse depositories in the world, and have significant experience in behavioral testing and in surgeries and injections of mice. So the phases after that, clinical testing, that is where we do testing on actual patients, and that would come after several rounds of discussion with the FDA on the safety and efficacy of our proposed treatments. And then if we get through clinical testing, then the idea is that we would make this therapy available to as wide an audience at as low a cost as we can, which involves trying to protect the intellectual property rights and raise as much capital as we can to carry this therapy through to delivery in patients through a prescription as we can, knowing that we probably need some help along the way with costs. So here is the approach that giant axonal neuropathy took. Now, giant axonal neuropathy is one of the diseases that is now in clinical trial at the NIH and has been since 2015. This disease is one of the diseases that Stephen Gray, our principal investigator, has been working on for the last few years, and he's taken them all the way through the process, from good lab results to this trial at the NIH, and is showing some very high levels of success in that disease. But you can see each one of these steps along the way. You need to see good lab results, both in a dish and in an animal. And then you need to start talking with the FDA about pre-IND. And IND stands for Investigational New Drug. That's what our therapy is an investigational new drug. And we need to discuss with the FDA how to approach the delivery of that and the testing of that to make sure that it's safe and effective in patients. These are the steps to do that. Model the results of the dosage and test to see if it's toxic, and then design a clinical trial using various oversight boards as sound boards and folks that would make sure that the delivery of this drug is safe and effective in our kids. But that takes time. So in this case, it took 7 years in giant axonal neuropathy. We think that following in the footsteps of diseases like giant axonal neuropathy, we could do it faster and cheaper. And I think we've proven that in this first stage, that we are on a faster and cheaper path. But it's still not going to be cheap and quick. So who is leading this? So this, as I said, is a parent-led initiative, but it's a parent-led initiative that's been designed and driven by QAHC, and other institutions and foundations are now on board with the science and with the fundraising to get us to where we need to be as quickly as we can. So this is very fast becoming a highly collaborative effort that is in large part being led by QAC. We're being led by a group of scientists too. The person on the top of that totem pole is Dr. Stephen Gray, who has been highly successful and is probably the most sought after person in AAV-mediated gene therapy for rare diseases. He has a lot of experience already, although he's a very young man, but he has a lot of experience already with 50 peer-reviewed papers in all of the top journals, and success with multiple diseases. His most recent with giant axonal neuropathy. So he has been involved in many different diseases and has as much expertise as anybody in this area. But he is only one part of a collaborative team. These are the folks, the major folks, and there are quite a few more, but these are the major folks that are involved in this project right now. So 9 people, all highly experienced. Steve Gray, I've talked about a little bit. His lab currently has more than 18 rare diseases at different stages of gene therapy development. We also have Natalia Morsch, who I talked about a little earlier. She comes from the NIH and has worked a lot with animal models of disease and rare disease and knows AHC as well as anybody. Dr. Neil Hackett, who's been in genetic medicine for more than 23 years. He's a direct consultant to us and working many hours per month to make sure we're on track. Nolan Sondhi, who has also more than 20 years of experience at Cornell School of Medicine, and she is treating multiple diseases now using AAV-mediated therapy. We have Steve Clapcoat on board, who's already rescued the symptoms of AHC in mice using a method called transgenesis, which can't be used in humans, like I said earlier, because it's too dangerous, but it proves the concept. Dr. George, who is at the Northwestern University School of Medicine, who has developed what's called iPS cells, induced pluripotent stem cells, from actual patients with the disease to test whether or not our viral vectors, our therapy, will work on specific patient cells. And I think that will be very important as we decide who will be eligible to participate in the clinical trials. Kevin S., who works a lot with him and has been instrumental in producing those iPS cells. Karen Lecky-Hartman, who's at Aarhus University, where they won the Nobel Prize for finding this protein. She has done a lot of work on mice with AHC, and we are discussing at the moment her participation in publication of papers associated with the data that we're generating in the experiments we're doing both now and in April. And then Kathy Swoboda, who is one of the top clinicians in the world, who is helping us to design our mouse experiments and also our clinical experiments, and is providing data associated with clinical evaluation so that we might be able to get through the pre-IND discussions with the FDA. So that's the core team. I think it's the core of some of the best scientists in the world associated with AAV and AHC. And I think it's indicative of a highly collaborative approach to a rapid generation of therapy for our disease. Here is the pipeline of what Dr. Steve Gray is working on right now, and I've obviously circled AHC as one of the diseases that he's now working on. We're in the discovery phase, and we've got a long way to go, but you can see that we're following in the path of diseases that have a very established track record, and some that have been highly successful in their AAV-mediated therapies. So perhaps I'll stop there before I get to the financial burden for any thoughts on the team or the approach or questions on anything else.
We don't have any other questions listed in the chat box presently, so if you have any, please do put them in there or send an email, jeff@curehc.org, or text 919-412-1870. I guess we can go on then, Simon.
Okay, perfect. So the financial burden. We have, I've broken this down into two phases. You'll see phase 1 on the right there is the proof of concept, and phase 2 is the path to clinical trial and beyond. Phase 1 is really what we're focused on right now. We've raised quite a lot of capital already for phase 1 and have started the experiments on up to the point where we are injecting mice that have the disease. So the first approach was, the first step of phase 1 was to generate a mouse model of the disease that is available to scientists around the world as needed to test various aspects of this proposed therapy. We managed to get that for free from Jackson Laboratory using NIH grant funding, and that should be finished being developed by April. We're using the D8-1N model, which is the mutation that is most common in AHD patients. It represents about 40% of all of the cases that have an ATP1A3 mutation, and has symptoms that reflect very well in mice what is represented in the human condition. So that should be done by April. The next thing was to design the viral vector for the trial in the mice. That's been fully paid for and is now ready for injection. It's being shipped today to Jackson Laboratory for injection on November 26th. And those experiments, we should see results before April so that we can pick our final vector for trial in diseased mice. The third step there is produce and test the vector for trial in mice. And that is going to cost two discrete sums. So about $120,000 to get us through each step of the process towards pre-IND and clinical trial, and that's being done by Dr. Steve Gray from University of Texas Southwestern. Another $80,000 or so will be towards what's called immunohistochemistry, making sure we understand where in the brain our vectors are going and how well they're expressing, and that should cost $80,000. And then the remainder is for the actual production of the viral vector therapy for injection in mice, and that's all being paid for. What we need to raise capital for now is the testing in the D8-1N mouse model, so the mice with the disease, and that we need $500,000 for. And to the extent that we want to keep this cheap or free for patients, we really need to raise this as a community. But I think probably the main thing that I'll stress here is in diseases like cystic fibrosis, you have an annual cost of somewhere between $350,000 and $750,000 per patient. Effectively, if we spend $750,000 here to test and vet our therapy, we should be able to keep that cost down significantly for our AHC patients. And the idea is that if we can prove it works, then we should be able to get industry to participate in the expensive part, which is phase 2, testing toxicology, getting a high enough quality therapy for delivery into humans, at large volumes, and then designing and executing a clinical trial. Those, we should be able to raise capital based on other diseases and their approach. We should be able to raise quite a lot of capital for our disease if we can prove that our thesis works in small mammals. So that's the idea. We want to be parent-led so that we can get there fast and focus very specifically on what our children need, and we'll get through phase 1 by doing that, and then provide incentives to industry and institutions to help us to get to the finish line by making that a competitive process so that we can keep costs down and timelines rapid. I think that's it, Jeff. I don't think I have anything else, but I'm open to questions.
Well, thank you very much, Simon, for doing that. I think, again, to drive home a couple of key points, I don't have any slides to add here, but as we're waiting for others to answer questions, I do want to drive home a few quick points. Just to know that with the collaboration between CureHC and other foundations, we have gotten a good chunk of that, or most of that part of the first couple line items on that chart there, the 30,000 to 220, already taken care of. And we've actually started to make a dent into that $500,000 figure as well. And really, the multiplying effect of many people doing little things adds up very quickly. I mean, imagine that we know of about 1,000 families in the world. If each of them found a way to raise a few $100s, we'd have this nailed fairly easily. And there are many easy ways to raise a few $100s in terms of fundraising. We do have resources through CureHC. People that have done fundraisers before that are willing to coach you. Different types of fundraisers, the ideas that we have done successfully that we can help you with. And this will potentially also, that's a good question here too. We have one other question. Someone's asking on the potential not only to help kids, but also to help adults with AHC. Simon?
I actually put the slide up here to show you the efficacy of AAV9. It works better, and this is one source, but so it's I think it's just one paper that has presented this, but it shows even higher efficacy in adults. Now we've designed our mouse studies in a way that it would test juveniles and young adult mice for the correction of symptoms, and for various reasons. So firstly, the brain cells will be more fully developed by the time that they are 4 weeks old or juveniles. And we think that that represents the likely clinical trial for humans, where we would not have neonates being tested for rescue. We'd probably have juvenile kids or young adults that would be in these trials. We also don't have prenatal screening for ATP183 mutations at this point, not widely spread at least. And the patient population is probably not going to be very young. So the idea here is that if we're going to treat people and kids that already are living with the disease, we really need to come up with a therapy that addresses their symptoms at their appropriate age. So the goal is to address this in young children, and also kind of kids up to teenage years. We are very likely to get adult participation in the trial, and it's just very hard to predict the level of success, even in the youngest kids, but especially in adults. Now, I will say, and again, I'm not a doctor, so this is just my opinion as a parent that's been deep into this. But my opinion is that we have an episodic based disease, and that to the extent that neurons have not atrophied to the point where they are no longer functioning, then functional neurons may be able to be rescued. And if we can rescue functional neurons, then perhaps we can prevent episodic attacks. And if we can do that, perhaps we can rescue some of the symptoms of the disease and prevent any further damage to brain cells or degeneration.
So, and we'll put that question to some of our medical experts. I imagine they'll have a similar answer to Simon's, but we will pose that question to them and get a
and I
estimate from them.
probably should stuff. So you're not going to get a response from scientists on that point. They're going to tell you that it's more likely to be to rescue younger kids than older people. But you're probably not going to get much more than that. I've asked many, many times. So that's why I led with my opinion rather than...
Gotcha. Gotcha.
Hey, Simon. At that point, I think we should talk to maybe the assistant director of FDA for orphan disease. They still call it orphan. I don't know why, but rare. They can give you a lot of guidance, and if you want, I could set that meeting up whenever you guys want, but it does take about 2 months to schedule. But they got a lot of insight on what we're doing, what we're doing, and understanding, what group they would like to see as far as, the trial for approval.
Yeah, and that's a great point, Lee. This is a very iterative process. So we as a patient-led organization would have our opinion. Our scientists that are leading the experiments would have their opinion, and the FDA would have their opinion. And we all get in a room and we hash it out. And
Yep. And we can set that. The first meeting can actually just be a call and just get a feel. But at some point, scheduling something up in Silver Springs and going in and meeting with them. And the guy that I met 3 years ago that was the assistant director is really pleasant, I think, to work with, and really open and understands all this. And I think he would take into consideration all that stuff. But I think always starting those discussions. I mean, if you're going for an approval, then obviously starting those discussions, earlier than later is better. So, like I said, whenever you guys are ready to get a feel for that, and I think we're a ways away from that. I think maybe this summer might be a time to just touch base and see if they have any issues with the AAV 9 delivery system and our approach. And I don't think they will, but it would start opening the door. And then as things progress, things get closer to a phase 1 human study, then you're definitely, got their feedback. So whenever you guys are ready to do that, I can, help you guys set it up. So.
Thanks, Lee.
Yeah, thank you. We have a lot of feedback already from the NIH. I know that the next step is pre-IND, and we're designing our pre-IND approach right now with Steve Gray taking the lead.
Yeah, we do also have a question. Can you give us a simple elevator pitch type of explanation to help share what's going on with others who may not be in the science-related field? I don't know if that's something we can do right here, or if we want to try and produce that offline and send that out to everybody.
You want to take a crack at that now, Jeff?
You want me to take a crack at that? I can certainly try.
Yeah, you take a crack, I'll take a crack, and then we'll do something official.
Right. Excellent. So I think what you can say to people is that what we're trying to do is introduce, AHC is caused by genetic mutation. We are trying to introduce healthy copies of that gene into the right places using a virus so that we can restore the function, hopefully effectively across the board, to eliminate all the symptoms and restore healthy function. There are a lot of steps that have to go into that process, and that's where those create money. We are trying to prove the concept in an animal model, which is the first step, and that costs a certain amount of money, and then you can use that particular chart that we had. And I think that's really all you need, besides talking about what those symptoms look like. And that's something you could do, either showing a video of your child or using the brochure we already have from CureHC that talks about the symptoms in terms of other diseases. And I think from there, that's what I plan to do individually myself. So Simon, do you want to add, subtract, change anything that I said?
Yeah, so maybe if you want a really short elevator pitch, and I'd say exactly what Jeff just said, but if you want a really short one, we're copying what's worked very well in very similar diseases, and we are testing it on small mammals to see if it has the effects we expect. And if it does, we think we have an approach to correcting the dysfunction associated with AHC mutation.
Beautiful. I think between those two, we might have something fit to print. Any other questions? Okay, well, I do want to take a few moments to thank Dr. Lee Mack, who is a member of our board of directors, for providing the platform for this today, and his excellent comments there towards the end. I want to thank everybody who's attended here today and for all the great questions. And I especially want to thank you, Simon, for help being the part of the principal driving force behind this, and for doing the presentation here today. We are greatly appreciative of all your efforts. As far as the recording, we will have that recording available. We'll send out an email with a link. The presentation we may send in a downloadable format through a service called WeTransfer, because it is a rather large file. I believe it's 16 megabytes, and so some email platforms may not allow something of that size, so we'll use a transfer protocol called WeTransfer. That gives you a link that you'll be able to download it from that way as well.
Hey guys, I wanted to jump in real quick. I kind of got in here late because I'm actually working.
No problem.
But I have a quick question.
Sure.
I had missed the presentation, but I'm going to catch up on it when you guys have it downloaded.
Yeah.
I have just a quick question. My son actually doesn't have the 1A3 mutation. He has the 1A2 mutation.
Right.
So I was just kind of seeing what kind of things you guys are looking at as far as, we're going down this
right.
1A3 mutation.
Right. You may have missed one little part of while we were in the midst of this presentation, Stacy, is that we can target certain parts of the brain depending on the particular promoters and the particular gene that we use in here. So if we prove the concept in ATP1A3, we certainly could look at doing a similar project for ATP1A2. I believe the mouse models of ATP1A2 are going to prove that mouse models are a good model for the ATP1A2 symptomology. And then we can certainly target the specific cells that ATP1A2 expresses in. It's just a different set of, it's obviously a different gene and a different set of promoters. And Simon, correct me if I'm wrong in that assessment. Simon, are you?
Just took a little while to get. Yeah, I'm here. Sorry. So that's correct. What we're trying to do here is we're trying to get a plug-and-play model for multiple different dysfunctions, especially associated with the ATP1 diseases. So we've got 10 different diseases in ATP1A3. We've got multiple in ATP1A2 and ATP1A1 also. So the idea is we've got an immediate set of genes that are of very similar size and structure and function that we can address not simultaneously, but perhaps consecutively. And we really should be addressing more diseases than just AHC if this methodology works. So that's the quick answer. I guess it's a slightly more controversial answer, but the dysfunction associated with the mutations in the sodium-potassium pump, so across ATP1A1 through ATP1A3 or ATP1A4, all of them are similar. Or not all of them, but many of them are similar, where what happens is the pump is not transporting enough sodium out of cells, or not transporting enough potassium into cells. And that dysfunction may be able to be addressed by delivering additional functional copies of the gene. So ATP1A3 looks very much like ATP1A2, which looks very much like ATP1A1. There may be ways to experiment with delivering ATP1A3 to glia, for instance, to correct for the high intracellular sodium in glia. But that's much further down the line than designing an ATP1A2 related treatment for glia.
All right. Okay, thank you. I know you guys mentioned something about videos. Are you guys interested in videos as far as kids' episodes? I mean, I know mine has it too, so it probably doesn't really relate with the videos. He has similar stuff that happens. It's just I don't think his is quite so severe.
Certainly, yes, you can share some of those either on the Facebook page if you're comfortable with that, or you can send videos to us through the info@cureahc.org email.
Okay. Okay.
And then if not, if they're too large to send via email, you can just email us and we can arrange to get them.
All right, Jeff, I actually have a YouTube channel that I posted some of Zach's videos on.
Okay. We will
So
we'll look to see that too. Excellent.
yeah, so I don't know if that's easier, and then that way, if you guys need them, you can grab them, or if not, then.
Thank you much.
Okay.
Any other questions, comments? Then what we'll do is we'll go ahead and conclude our time together. Again, thank you everyone for being on the call. Thank you for all the questions. Thank you, Simon, for all your hard work.
Thanks so much, everybody.
Take care.